bond. Now, why do we not use glycogen as our main energy store? It's because it has a variety of reasons, but the first one is a limited reduction. So remember, reduction here is talking about redox and oxidation, so it has limited rounds of reduction. It doesn't yield as much ATP as the complete reduction of fatty acids, and we'll talk about that in the fatty acid lecture. But fatty acids can go through many more rounds to generate ATP. But it is readily metabolized. And so this is a ready source. It's an easy, quick source, whereas fatty acids are more of a longer-term source of energy. And it can be used in anaerobic conditions. And that's important because under exercise, where oxygen levels might be reduced, you need to have an energy source. And for fatty acids, that's not going to be possible, so we use glycogen. And because of this reduction limit in ATP, we just don't have the store. So you can see, as we'll get into later, we just don't have the same amount as we might have for fatty acids. Now, as we saw with the learning objectives, we're going to talk about liver and muscle. And the reason for that is that they're the major stores of glycogen. So liver and muscle is the major location where we'll find glycogen. And again, this is either to control the level of glucose within the blood, so for the liver, and in the muscle, it's really for that activity. And it constitutes 10% of the weight of the liver versus only 2% by weight in the muscle. So you can see you've got a very limited supply in the muscle. This is going to be expended very quickly with exercise, and then you need to use alternate sources. And as I mentioned before, the liver serves to maintain blood glucose levels so that you have a homeostatic control. And skeletal muscle, the glycogen there is to serve the energy demands of muscle on demand. And so this is when you've got a fight or flight response, as an example, where you have to run, then you need rapid energy. Now, in terms of the storage, glycogen exists as small granules within the cytoplasm. The size doesn't really matter, 10 to 40 nanometers, but very small. And they each contain about 55,000 glucose molecules. So it's very, very densely packed, and that's an important concept. And this dense packing really comes about with those bonds. I've highlighted this again, we've mentioned it before, but whenever you see something in bold, just to highlight the importance, in the liver, glycogen synthesis and degradation are regulated to maintain blood glucose levels. So this is a key theme that we will talk about. The level of glycogen within the liver, if you've got too much glucose, you're going to try and build up more glycogen within limits. And if there is a demand for glucose, this is going to be a quick and rapid release mechanism, so the degradation of glycogen to release the glucose, which can then be used for energy. So this diagram here, this is showing, first of all, the branch structure, so a very gross overview of glycogen. Now, here you've got a letter G, so as it says here, a protein called glycogenin. This is important for the formation or the initiation of this cluster, so you only have the one protein and then you have all of the other bonds. Remember that most of these that are linear are alpha 1, 4. These points of junctions where they branch off are alpha 1, 6 bonds. So structure of glycogen now shown here. So each one of these individual units is glucose. But because this is a polymer, these are then joined together, and you can see that the most common bond is going to be the 1,4, and we see it here. So one position on this glucose molecule joined by an oxygen atom to the 4th position, and if we go to the 6th position, and the 1,6 across here. So this is where the branching occurs, and this is the linear profile. You can see here the two ends, the non-reducing ends. This is really just about the points of reactivity, but this is going to go from left to right, and it can extend. Now, glycogen degradation. We mentioned up here, the liver, glycogen synthesis, so making it, but degradation as well, maintains blood glucose levels. So what is glycogen degradation? It's glycogen lysis or glycogenolysis. So whenever you see lysis, it's cutting, and we'll talk about it multiple times, but we will talk about lytic events or lysis, particularly with fatty acids, because with fatty acids, we have to chop them off 2 carbons at a time. So this is where you'll see it a lot, but it's the same idea here. Glycogenolysis, where you're starting to break the glycogen, into its glycogen or glucose units. So breakdown of glycogen yields glucose 6-phosphate. So not glucose on its own, but glucose 6-phosphate, and this is an important addition for activity. Now you might think, well, okay, we just need to cut the bond and off we go. Actually requires 4 enzymes. It's a more complicated process. The first one is to degrade the glycogen, which is this first part. The other 2 to maintain it as a substrate, because it needs to be metabolized. And then 1 to convert it into that suitable form for metabolism. And this last point is also very important. Whatever your precursor molecule is, it needs to be in a state that is able to enter the metabolic pathways, so that whatever energy, whatever ATP it's able to release is in a form that is compatible. If it's not, then it can't enter those pathways. You're not going to generate that ATP. So very, very important concept. So let's go through these. So the first one to degrade glycogen. So we have a phosphorylase, cleaves glycogen to release glucose 1-phosphate. So this is the enzyme that is able to alter the metabolic pathways so that whatever energy, whatever ATP it's able to release is in a form that is compatible. If it's not, then it can't enter those pathways. You're not going to generate that ATP. So very, very important concept. So let's go through these. So the first one to degrade glycogen. So we have a phosphorylase cleaves glycogen to release glucose 1-phosphate. So this is the enzyme that is cutting or leading to that lytic event. So glycogen phosphorylase is the key regulatory enzyme of glycogen breakdown. As I mentioned in previous lectures, whenever we talk about pathways, those key molecules, the rate limiting step, the initiating molecules, the initiating enzymes are critical to your understanding. So this is one that you must know, because really it is the first one that allows you to go from glycogen to glucose, and it also is the key regulatory point, so it controls all of those downstream effects. So this is a really, really important enzyme. So it cleaves glycogen at that 1,4 position. Could have put alpha, but we know it's the 1,4 position. And it does this by adding orthophosphate. Now, orthophosphate is actually 2 phosphates, and this breaks the bond to, orthophosphate is 1. I was thinking of pyrophosphate. So orthophosphate is 1 phosphate, and that leads to glucose 1-phosphate. Look, it's on a different position because we want a glucose 6-phosphate, so make sure you understand the difference here. This is just to release it, but remember it's not in the state that we wanted, which is why this last enzyme at the bottom here is required. So glycogen plus phosphate, inorganic phosphate. We have lots of phosphate within the body. You know that ATP has 3 phosphate molecules, so there's a ready source available. This converts or splits one of those glucose molecules to glucose 1-phosphate. We've now got the glycogen that continues, but we say N-1 residues because we have lost our residue. We have lost one glucose was the major is the key regulatory enzyme of glycogen breakdown. As I've mentioned in previous lectures, whenever we talk about pathways, those key molecules, the rate limiting step, the initiating molecules, the initiating enzymes are critical to your understanding. So this is one that you must know, because really it is the first one that allows you to go from glycogen to glucose, and it also is the key regulatory point, so it controls all of those downstream effects. So this is a really, really important enzyme. So it cleaves glycogen at that 1,4 position. Could have put alpha, but we know it's the 1,4 position. And it does this by adding orthophosphate. Now, orthophosphate is actually 2 phosphates, and this breaks the bond to, orthophosphate is 1. I was thinking of pyrophosphate. So orthophosphate is 1 phosphate, and that leads to glucose 1-phosphate. And look, it's on a different position because we want a glucose 6-phosphate, so make sure you understand the difference here. This is just to release it, but remember it's not in the state that we wanted, which is why this last enzyme at the bottom here is required. So glycogen plus phosphate, inorganic phosphate. We have lots of phosphate within the body. You know that ATP has 3 phosphate molecules, so there's a ready source available. This converts or splits one of those glucose molecules to glucose 1-phosphate. We've now got the glycogen that continues, but we say N-1 residues because we have lost our residue. We have lost one glucose molecule, which is shown here. And this process is called phosphorylysis or phosphorylysis, because we've got a phosphate group and we're lysing. So it's the same idea, it's just adding the phosphate as the molecule, which is breaking the bond and releasing that particular glucose molecule. So the phosphorylase, which we've got up here, glycogen phosphorylase, leads to the sequential removal of glycosol residues. Glycosol is the correct term, but you could also just say glucose residues because remember it's just a string of glucose molecules. These are removed from the non-reducing ends of glycogen. So these are those ends with a free hydroxyl group. So here is a glycogen molecule. We can see here we've got a free hydroxyl group. Here there is no free hydroxyl group because there's going to be a bond. So this is the free one with N residues. So here it's 4. Here's the phosphate coming in. Obviously the enzyme is going to be acting. We add it to this bond here to break the alpha-1,4 glycosidic bond, and we see the release of glucose 1-phosphate because remember this is 1 to 4, so this is the 1 position, and we then have the glycogen residue which has been released, and it's N-1 because we've lost this particular glucose. If the cell needs more, what does it do? It just keeps going, chop, 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 keeps going until it needs to remove that molecule. Now, glucose 1-phosphate, we don't want that, but not surprisingly, it's readily converted to glucose 6-phosphate. Makes it very easy for the cell to use. This is the preferred and the required substrate. It's energetically favorable because we have this phosphate group. And what you can think of here is this is very similar to what we see with ATP. When we talk about energy within the cell, we talk about ATP. Where is the energy? The energy is in the bond with the phosphate, and releasing that phosphate releases energy. And in essence, we've got the same thing here. We've got a phosphate group attached to the glucose. Releasing it, moving it, because we're going to have to move it to a different carbon, is going to release energy, which makes this thermodynamically or energetically favorable because of that phosphorylation state. So it's going to facilitate this process. So one step facilitates the subsequent step. What else do we need? We need a deep branching enzyme, and this is required for glycogen breakdown. Remember that glycogen is a highly branched molecule. So 1 in 10 residues of glycogen are branched. Glycogen phosphorylase cannot cleave at the alpha-1,6 bond in glycogen. So recall before we said the phosphorylalysis occurred at the alpha-1,6 bond, alpha-1,4 bond, but we have alpha-1,6 bonds also being present. So how do we work at that particular location? So what we see is that the glycogen phosphorylase actually stops lysing at 4 residues away from that branch point. So if we say that there's 10 residues, well, 4 away means that only 6 molecules of glucose would be released. It's a great reduction in the total capacity that could be released, so there has to be another way. And what we see is that the remainder of the glycogen is degraded through a process called remodeling. And this is basically just shifting the groups around so that the enzyme is able to continue its activity. So remodeling and activity of a transferase. And the transferase, as its name suggests, transfers groups. And there is also an alpha-1,6 glucosidase. So these are the 2 enzymes that are important in this remodeling process. What does it do? The transferase shifts a block of 3 glucose residues, remember, glucosol, glucose, interchangeable term, from one outer branch to another. It removes those last 3 and shifts them elsewhere. And by removing them, there's now a continuation of that linearity, and the enzyme is able to continue to release those individual glucose molecules as is required. And so what this does is that it exposes a single glucose residue which is linked by an alpha-1,6 linkage. That should be alpha, not A. Apologies. And this is hydrolyzed by the alpha-1,6 glucosidase. So remember, we had the second enzyme that is able to cut at that location. And that free glucose is now phosphorylated by another enzyme which is called hexokinase. You can tell what this is going to do. Hexo is telling you 6. Glucose is a 6-carbon molecule. It adds phosphate. A kinase is an enzyme that will add phosphate. So 6-carbon phosphate adding molecule. And in this context, we know that that substrate is going to be glucose. So if we look at this diagram, here is a string of glycogen. So we've got our alpha-1,4 linkages in a line, and it's saying core because this is where it's linked to the protein, and this is the first branching point. And then we've got another string of alpha-1,4, and here's the alpha-1,6 linkage. You want to remove those glucose molecules. We've got lots to cut off, and so here we're showing 8 that are being removed, so there's 8 phosphate molecules, just showing you as a collective. You could repeat this 8 times, obviously, but it's just showing you as one, and this is where the phosphorylase comes in. We now have a branching point, and we've got 4 glucose molecules, and we said, well, when we've got 4, phosphorylase is not able to work. So what happens? Transferase comes in, it cuts this bond, and it shifts it to the end of the previous grouping. And so if we see in this diagram, these 3 are now attached to the yellow glucose molecules here. It's taken it and physically moved it across. And so we've now got 1 glucose molecule here. Now this could be problematic, but remember, we've got an alpha-1,6 glucosidase. It acts on this, chops it, and removes it, and we now have this string of linear alpha-1,4 glucose molecules. Phosphorylase is able to come along and just cut, and we've no longer got that issue. So it does require some additional steps, but you can see limited requirement in terms of activity. By removing that group, you've got this massive linear glucose chain, which allows for the enzyme to be active and in turn release all of those molecules. So as it says, diagram showing the glycogen remodeling, action of the transferase, removing 3 glucosol units, so 3 glucose molecules in blue, 1,6 glucosidase, which hydrolyzes, so hydro referring to water. You can see why I've got water molecule here to remove that last glucose molecule. This diagram is now just showing you that hydrolysis. We've shown it simply here, but here is another way of demonstrating this. So here's glycogen, we've got that bond. This is the 1,6 bond because this is what we're looking at here. You've got the water molecule, you've got the alpha-1,6 glucosidase. Based on the nomenclature here, we know that this is a 1,6 bond because this enzyme would only work on a 1,6 substrate. This gets broken, and you can see that what you're going to do is that oxygen is going to be converted to a hydroxyl, and you've got the same thing occurring on this side here. So we've got glucose and the glycogen residue as well, but it's now shortened by 1 glucose molecule, and this can then go off and be used. What else do we need? The third one, phosphoglucomutase. Phospho for the phosphate, gluco for the glucose, and mutase. Mutase can be thought of as a converter. It converts it from one state to another. That's all it is. So this is taking glucose with a phosphate group and converting it from one form, so glucose 1-phosphate, which is what we had released initially, and converting it to glucose 6-phosphate. In essence, is shifting the position of the glucose from the 1 position to the 6 position. That's all. As I've written here on the second line, a mutase is an enzyme that shifts the position of a phosphate group on a substrate, and it contains a phosphate group itself. So the enzyme contains a phosphate group on a serine residue, and the serine residue is part of the active site. Now, why is this important? This phosphate group on the serine is involved in an exchange reaction. So it is exchanged to the glucose 1-phosphate, and the 1-phosphate is then removed. And in turn, it's returned back to the enzyme, and this now leaves the glucose as glucose 6-phosphate. It's essentially swapped over. The phosphate that is on the mutase enzyme is transferred. The 1 position is removed, given back to the enzyme, but is now transferred onto the 6 position. So it's a bit of a rearrangement. And by transferring the phosphate back to the phosphoglucomutase, it retains its activity. It's able to repeat the next cycle. We've been able to shift the phosphate from the 1 position to the 6 position and now convert the glucose into a substrate, which can be used metabolically, which can be used for enzyme action and, of course, for energy production. So this is what this is showing. What we can see down here on the bottom, we're in yellow, this is the phosphoglucomutase. It's just showing you a very small portion of the active site. And you know that the active site is going to be a protein, and proteins are made of amino acids. And here we've got serine as the amino acid with the phosphate group attached. Here is our glucose 1-phosphate coming in as the substrate. And here is the phosphate on the incorrect position. Incorrect for metabolism. You've got glucose 1,6-bisphosphate. So this phosphate gets attached to this group. So that's the first step. So the mutase donates it. It's 1,6-bisphosphate because we've got 1, 2, so biphosphate, and they're on the 1 and 6 position. This is no longer required, so it gets donated back to the mutase, and we can see it's now brought back to this location. And in doing so, we've been able to convert the enzyme back to its preferred state, glucose 6-phosphate state, which is shown here. This is now able to go into metabolism and can be readily used. So I'm going to stop there simply because I think it shows really nicely how glucose can be released from glycogen, the importance of
Okay, so we've released the glycogen, so now we've got to talk about the release of glucose from the liver. And as we've said now multiple times, major function of the liver is to maintain a constant level of glucose in the bloodstream. And this is important for homeostasis. The phosphorylated form of glucose is not transported out of the cells. The phosphate interferes, probably through a charge interaction with the transporter, and that interferes with its ability to be released. Now, we know that the liver contains the enzyme glucose 6-phosphatase, and we saw this in the prior lecture with gluconeogenesis. And this is a hydrolytic enzyme. And what it does is that it removes the phosphate group from the glucose 6-phosphate. So you can see that here, very simple reaction, glucose 6-phosphate, which is what we had at the beginning, through hydrolysis gives us glucose and the free phosphate. That glucose can now leave the liver and enter the bloodstream. So we've got storage of glycogen, and we said that this is mostly in the liver. We then have glucose 6-phosphate being produced, but it can't get out into the blood. And so what we now do is we remove this group, and this is now able to exit and go into the blood. That's all that we're talking about. It's a very simple sort of concept. Now let's talk about the regulation process, because the release so far I think has been fairly simple and straightforward. So let's think about the phosphorylase activity. So glycogen degradation is controlled by several mechanisms, and these all focus on glycogen phosphorylase. So this is going to be the key enzyme that we need to focus on. It is controlled by several allosteric effectors, and these all signal the energy state of the cell. And what we mean by this is if the energy is low, energy needs to be replenished, and so we want the glucose to be released, so ultimately we get ATP. If ATP levels are high, the drive to make additional ATP is not there, so the reverse would be true, that we don't want the breakdown of glucose, and therefore we don't want the breakdown of glycogen. So that balance is really quite important. So we've got these allosteric effectors. We've also got the ability for reversible phosphorylation. And remember, this just means that we can add the phosphate and we can take it away. And this response is mediated by different hormones. And you would have probably heard of most, if not all of these. Glucagon is one, adrenaline you would have heard of, and I know insulin because we spoke about this in the previous lecture. Now we're going to compare the control in the liver and the muscle, but we will do this separately. So make sure you understand what happens in the liver, and then when you get to the muscle, you can then compare and contrast the differences between the two. That will make the learning an easier process for you. So in the liver, the enzyme, and what are we talking about? We're talking about the glycogen phosphorylase, as it says up there. exists in two forms. There's an active phosphorylated form, so phosphorylase A, so A equals active, and there's an inactive unphosphorylated form, or phosphorylase B. Now, B doesn't lend itself to anything that tells you that it is off, so if you remember that A means active, then you know that B means off. And as we saw previously, we said that the active state refers to the R state, and the less active state is the T state. So in the liver, it exists mostly in the active A form, so phosphorylase A or the R state. Don't get caught up in this terminology. We're talking about the same thing. And to be honest, if you just remember phosphorylase A and you don't worry too much with the R state, I think I would be happy with that. So by doing so, we're going to get the conversion, we're going to make glucose, and this is going to provide glucose for the whole body. The binding of glucose to the enzyme changes it to the off state. So glucose is itself an allosteric modulator. Okay? The substrate can modulate the activity of the enzyme, and by doing so, it makes it less active because excess glucose must be present if this is occurring. And so therefore, there's no need to degrade the glycogen. So this is a nice control mechanism that takes place. In contrast, if we look at the muscle, what we see is that in muscle, the default state is actually the inactive B form, or the T state. So this is the off state. And this makes sense because in muscle, you don't want to be breaking down glycogen if the muscle is resting. It's only when it is active do you want it to do that. So only activated during muscle contraction. And this is where you'll see the shift between the B and the A form. It's also activated by high levels of AMP. And again, remember what we said, ATP goes to AMP and phosphate, or phosphate itself. So AMP, if this is high, it means that the level of ATP is low. So more energy is required, and it makes sense then that we would want to activate this pathway. And similarly, where we said glucose can block it, here we can see that glucose 6-phosphate can actually stabilize the B form. It will turn it off by keeping it in the off state, and this prevents excessive breakdown. So phosphorylation converts phosphorylase B to the A form, and it occurs at a specific serine residue, and it is initiated by various hormones within the body. So low blood glucose leads to glucagon expression. Exercise releases adrenaline. So we've got the two regulatory molecules that are occurring. So these lead to phosphorylation and hence activation of the enzyme. Now, how does this signal get received for glycogen breakdown? No, it should be from, not form. So from glycogen breakdown. There is a kinase called protein kinase A, PKA, and we'll talk about this in more depth later in a subsequent lecture on signal transduction. But all you need to know is that this is a particular kinase that is very important, and it is the one that is responsible for phosphorylating the phosphorylase. So again, the B going to the A form, because remember, this is the off state and this is the on state, so we want to add the phosphate. So what is the cascade for this response? Now, let's have a look first of all at the hormonal process. This is one of the diagrams that we haven't yet covered. So diagram showing the hormonal control of glycogen breakdown. If we look at the diagram, there is a left side and there is also a right side down here. So the left side shows the hormonal response to fasting, as we see here, so low glucose. So glucagon stimulates glycogen breakdown in the liver. Glucagon will want to stimulate the breakdown of glycogen because glucose is low and we want to increase it. So what we can see here, glucagon is released from the pancreas. In the liver, glycogen is broken down to glucose and it enters the blood. Now don't forget, particularly when we come to talk about it on this side, the Cori cycle. Lactate can be converted to glucose and that glucose can then go back into the muscle. Remember in the previous lecture we spoke about lactate being produced, a bit of a dead end we called it, metabolically. It goes into the liver converts it back to glucose, and then it can go back into the bloodstream and make its way back to the muscle. So these are all very much interconnected. On the right-hand side, it shows you the hormone response in response to exercise. So this is vigorous exercise. We have adrenaline being produced. So this is an American textbook or North American textbook, so it's referred to as epinephrine here. We would say adrenaline, same thing, just a different name. This is reduced from the adrenal medulla on the kidneys, goes into the bloodstream. And you can see here epinephrine comes across, binds to the transporters that facilitate this process of glucose coming into the bloodstream. So that enhances glycogen breakdown in the muscle and the liver to provide fuel for exercise. So again, if you look here, in the muscle, glucose is broken down to glucose 6-phosphate, pyruvate, and lactate because this is an energy-intensive process. The glycogen that is within the muscle can also be converted, and this in turn gives rise to that energy that is occurring. And remember, the lactate goes back into the blood. Now with this diagram, they're showing it going down, but really it's going down, it's going to come back up through the top and then go back into the liver. And you can see that with this diagram in terms of how it's mixing together. So this is linking in what we've seen previously, but now just showing how glucagon is affecting glycogen metabolism. Now, if we look at the signal transduction pathway, so diagram showing the signal transduction cascade activating the phosphorylase enzyme. So remember, B goes to A. So the hormones, glucagon and adrenaline, we've seen them in the previous diagram. How do they work? They activate a receptor. So you know that a hormone will bind to a receptor. That receptor leads to an increase in a molecule called cyclic AMP. So AMP, ATP can be converted to AMP, but then if you've got a bond that makes it more of a circular molecule, it's referred to as cyclic AMP. Cells are very sensitive to the level of cyclic AMP, particularly when it goes up. And this increase in cyclic AMP is what activates the protein kinase A. So the A in the cyclic AMP refers to the A in the protein kinase. So you can see when this goes up, this is going to become activated. So as it says here, this activates pKa, which activates the phosphoryl kinase. It phosphorylates the B form to the A form, and we start to get conversion. Now the structure of the molecule up here. So here is the receptor, epinephrine and glucagon. They bind to a receptor. The 7-transmembrane receptor will be covered in depth in the signal transduction lecture, so don't worry too much about it. But it has these subunits that essentially, when they get activated, act on this enzyme, which is called adenylate cyclase. And adenylate cyclase is the enzyme responsible for converting ATP to cyclic AMP. And as we said here on the right-hand side, protein kinase A is very sensitive to cyclic AMP, and the red is shown as the off, the green is shown as the on state. So that binding turns it on. The protein kinase now activates the phosphorylase, and the phosphorylase then acts on the enzyme to go from the off state to the on state and allows that process to occur. So this demonstrates the cascade and also demonstrates the linkage of activation of hormones and the transduction of signals either in the liver or in the muscle. So what is the point of this apart from starting the process? When you have a signal transduction process, it amplifies the initial event. So you could theoretically have one molecule of hormone, one protein, and this will give rise to multiple cyclic AMP molecules. And by doing so, it will set off the cascade. So the cell is very, very sensitive to just very small changes in hormone and is able to amplify that reaction. So what happens is that once the energy needs have been met, the phosphorylase kinase and the glycogen phosphorylase are both turned off. So again, think of this a bit like a switch. We have a need, we turn the switch on, we have this entire pathway becoming activated. When we reach a homeostatic level, we have to turn it off, where the opposite of activation for a kinase is dephosphorylation, and this is the phosphatase. So this leads to inactivation, and it also then leads to glycogen synthesis, and this becomes stimulated. Because if we've reached this point, there's typically going to be an excess of glucose, so we want to renew the glycogen that has just been broken down. This is a very active process. As soon as you break it down, as soon as the requirement ends, bang, the cell has to go backwards to try and reactivate the process. So this is quite an active, it's not a static process as you might think. Okay, let me stop there because we've covered a fair bit. Make sure you understand the principles between the liver and the